We have invited two speakers who are working in Scotland, who are part of this network. The first one is Daniel Wang. She's now in London, actually, but she's been working in Glasgow for quite a while. And the second speaker of the day is James Dowsett, who is working at the University of Stirling. And we can get started with the first talk already. Daniel, please tell us about theta and gamma entrainment.
So today I'm going to present you my postdoc work with Simon Hansen-Meyer about how theta and gamma entrainment something memory. And this work is also with contributions from my colleague Andrew Crowther and John Parrish, as well as my supervisor Kim Shapiro. Okay. So imagine how you spent your last New Year Eve. So you might celebrate together with your family, with your friends, with some music, lights, fireworks. And episodic memory is the mechanism that allows us to form, hold, and revisit such rich and often multisensory memories. Okay, so the puzzle is that all this sensory information is processed the specialized regions that distributed across the cortex. So how are they bound into coherent memory? The hippocampus has been suggested to be the key because it receives all the sensory information, all the information from sensory regions. And more importantly, there are two synaptic plasticity mechanisms that have been found highly active in the hippocampus. One is theta-phase plasticity, and which suggests that theta oscillations as the dominant hippocampus provide a time to dose for long-term situation or depression. So rodent studies show that stimulation at the opposing theta phases induces LTP or LTD. And the other synaptic mechanism is spike timing dependent plasticity, STDP, which suggests that whether LTP or LTD happens depends on the spike timing between presynaptic neurons and the postsynaptic neuron. If presynaptic neuron fires in a short delay before postsynaptic neuron, LTP happens. LTP happens in the order of firing is reversed. So the evidence supporting how these two synaptic plasticity mechanisms supporting learning and memory is mainly found in animal studies. So first, we investigated if the theta phase dependent plasticity applies to human episodic memory. If it does, then we expect when the visual and the auditory information arrives at the same time in the hippocampus, then associated memory should be better when the sensory information arrives out of theta synchrony. So to achieve manipulating the degree of theta synchrony between visual and auditory activity, we use this rhythmic sensory stimulation paradigm where we modulated volume of sounds and the luminance of videos at the 4 Hz, which is the theta frequency band. As well as we manipulated the phase offset between the visual and the auditory 4 Hz activity, which is 90, 180, and 270 phase degrees. And then we asked the participants to memorize the associations between the videos and the sound, and later we tested their memories about the association. So, as expected, the recall performance for the synchronous condition was significantly better than the asynchronous conditions. And later we also replicated this finding in an independent, entirely new set of participants, where we recorded the participants' EEG activity and sorted the trials according to their actual phase differences between visual and auditory 4 Hz activity. Okay, so next I'm going to show the evidence on the movement of the synaptic plasticity mechanisms in this theta-induced memory effect using a computational model built by my colleague George Parrish. So our model is a neural spiking model, and it includes neocortical and hippocampal systems, both with 2 groups of neurons. And the hippocampus receives the inputs from the corresponding neocortical neurons, as well as an ongoing theta oscillation at 4 Hz. And the hippocampal synaptic weights are modulated by the STTP learning rule. And more importantly, this STTP learning is theta phase dependent. So to simulate our experimental paradigm, the neocortex received 2 inputs of 4 Hz sine waves. And the phase modulations are like what we did in the experiment, which the one sine wave was phase offset on the other sine wave with 4 phase offset, 0, 90, 180, and And to evaluate the recall performance of we check the weight changes after learning from auditory group of neurons to the visual group of neurons. So first we show that the weight in the zero phase condition was better than the 3 asynchronous conditions, and the weight in the 3 asynchronous conditions differ from each other. So this replicated our previous findings and suggests that synchronized proteins increase associated learning. And secondly, it successfully replicated the findings from our previous study, which showed that this memory enhancement in the synchronous condition was specific to the theta modulated stimulus, but not the alpha and the delta modulated stimulus, because only when the stimulus modulated at the theta frequency, they coincide with greater LTP. And we also replicated the finding from a control experiment that shows this theta synchronous memory effect goes beyond perceptual finding, because this study showed that the memory performance in the theta synchronous condition was even better than the memory performance when the stimulus was not modulated. And we show that indeed that's the case in our simulation. Next, we experimentally investigated if the STTP is involved in human episodic memory. So we modulated the luminance and the volume of videos and sounds at 37.5 Hz because this frequency allowed us to precisely control the sensory stimulation peak delays between the two sensory stimuli. And yet, we can see here in the 90, 180, and 270 the peak delays between visual and auditory stimulus was 6.7 and 13 and 20 milliseconds respectively. And the other important characteristics about this STDP learning rule is the directionality of the association, because LTP only happens when the presynaptic neuron fires in a short enough delay before postsynaptic neuron, not the other way around. So in our experimental design, if we look at this image so they represent the peak delay from visual to auditory stimulus. For example, in the 90 condition, we could see that from the visual to auditory stimulus, the delay is the shortest. But if we look at the greenish bar, they represent the delay from auditory to visual stimulus. So it is actually the longest delay in the for the from auditory to the visual stimulus. So that means if we cue the memory with different modalities, then we would expect that the memory changes as a function of these peak delays in an opposite direction. So memory would become better along this direction, but memory would become better along this opposite direction. If, memory would become better along this direction if we cued with a visual stimulus, but memory would become better along this opposite direction if we cued with an auditory stimulus. Yeah, so that's exactly what we did in the experiment. So in experiment 1, we cued participants with video and asked them to recall which sound was paired with this video earlier. And in experiment 2, we cued participants' memory with one of the sound and asked them to recall the paired video. And then we simulated the result with our computational model that implements only the STDP learning. And we show that, so in this way we can make predictions. So we show that the synaptic efficacy in the 90 condition, when it is the shortest delay from visual to auditory stimulus, the synaptic efficacy is larger than in the visual cued condition than in the auditory cued condition, because it was actually the longest delay for the auditory cued condition. And the pattern is reversed in the 270 condition, because then it's the longest delay for the visual cued condition, and it's the shortest delay for the auditory. And so we computed the instantaneous phase differences between the visual and auditory activity, and we showed that, and we sorted the trials into 4 equal phase bins, and we show that the mean, the instantaneous phase differences between grand average ERP did show that a phase concentration in 0, 90, 180, and 270. And then we calculated the recall accuracy in each phase bin, and we show that the recall accuracy for the visual cued group was significantly larger than the auditory cued group in phase bin 2. And the pattern was reversed in the phase bin 4, which is consistent with what our computational model predicted. So further, we linked our empirical findings to the computational model by localizing the inter-trial phase coherence differences between subsequently remembered trials and subsequently forgotten trials. And we found that the maximum ITPC difference was localized in the medial temporal lobe, also including the hippocampus. And we showed that, so what we did to the model simulated results was that we compared the ITPC for the higher synaptic efficacy trials versus lower synaptic efficacy trials. And we also saw that the ITPC was higher for the high synaptic efficacy trials than the low synaptic efficacy trials. Okay, so in summary, we hopefully today show you that we can manipulate the sensory inputs with high temporal resolution, and the human episodic memory is affected by the stimulus inputs timing, probably via the two synaptic plasticity mechanisms. By showing that, we hope hopefully to bridge the gap between the rodents in vitro studies and the human episodic memory behavior. Okay, so yeah, I would like to thank my lab members in Glasgow and Birmingham, and as well as the funding bodies ESRC and ERC, and thank you for your attention.
Thank you, Dundee. So that was our first talk. Because of our format today, we don't really have time for questions from the online audience. So if you're interested in what Dundee is up to, and if you have any questions about the presentation, please do get in touch with her. She should be easy to find. Just look at her papers and her contact email address. Okay, so while we have talking, you might have noticed that we had to switch to a microphone because the in-room microphone doesn't work because of the storm that is going on. And during the first talk, we also had the first water coming into the room. So you might see the sea level rise live while you're watching the next talk. So I'm just handing over to James Dowsett at University of Stirling now, who's going to tell us about SSVPs and really cool glasses.
Okay, thank you. Thank you for inviting me. Okay, so I want to begin firstly by highlighting a few limitations, what I think are key limitations of the way in which we do cognitive neuroscience. And these are sort of real key challenges that I think as a field we need to address. So the first problem is it move to the next slide?
The other side, try the other way. No, press the bottom arrow. Yep.
Okay, thank you. Yeah, so the first of these key problems is movement. So movement is really fundamental to how the brain works. So from an evolutionary point of view, the real purpose of a brain, the primary purpose of a brain, is to move an organism around its environment. And although many of us sort of spend our lives now, like the figure far right, sort of hunched over a screen, all of the evolutionary pressure on how our brains evolve to function has been whilst moving around in the real world. And particular importance for our species is like 4 to 6 million years ago when we started walking upright on 2 legs, which placed very different demands on the visual system and the vestibular system. So in animal neuroscience, they're sort of way ahead of us. People are able to record from thousands of channels in freely moving animals, which is amazing sort of stuff's going on, to where are the freely moving humans. And here's the problem, of course, they freely move into our labs and we fix them. We fix people in chin rests and we get fixation crosses in EEG and MEG, and in the scanner people have to lie down. So the limits of our methods constrain the limits of the questions we can ask. So what we really need is to get ways of studying the brain whilst we're moving around. The second problem, which is kind of a related problem, is ecological mobility, which is that our brains deal with all of this complexity in the real world. And when we try and study the brain, we really often usually have very highly controlled artificial laboratory setups. And yeah, many different authors have sort of highlighted this as a problem, the problem of ecological validity. I think these authors in this paper have quite a nice sort of illustration of this, where I consider the difference between looking at a picture of a face on a screen in a sort of lab environment versus looking at that same face as part of a real conversation when you're actually talking to someone. All of the body movements, all of the posture, all of the eye contact, all of the social context, and all of the emotional salience is completely different from that very highly controlled situation. And the key point here is the underlying functions, neural functions, might be completely different in real world situations compared to these highly controlled lab situations. So the method that I use is, like the previous speaker, is flickering light, the SSVEP, which is really just the brain's response to visual stimulus, which is flickering at a specific frequency. So the way in which this has been done, sort of historically, has been one of two ways. It's been with LEDs directly into the eyes. In this case, this was in the lab where I did my PhD with Christoph Hermann, and this is with LEDs just directly flashing into the eyes. So this gives us a very strong signal, obviously, because it's very bright light. And this is the advantage that we can stimulate at any frequency. We can really flicker at any frequency. But it's not very interesting. It is just a flickering light. The other way, which is the far more common way that everyone's familiar with, is brain-computer interfaces, frequency tagging approach, where various elements on the screen can flicker at different frequencies, which is great because we can do frequency tagging. But you are limited to the refresh rate of the screen determines the frequencies you can actually flicker at, and frequency is important. And also, you have to be looking at a screen, so you are limited to these sort of controlled artificial situations. So the method that I'm working on, and I've got a pair of these glasses here as well, just for people in real life. But yeah, these are LCD glasses. They're originally designed for 3D TV. And what I've done is adapted these glasses so that they can flicker at any frequency. So whenever any voltage is applied across the LCD glass, they go dark. So not completely dark, but like dark like sunglasses. And we can drive these at any frequency. So we can flicker light and dark at whatever frequency we're putting the current across the glass. These glasses have three key advantages, I think. So the first is that we have SSVEPs of real-world scenes, real-world objects, and real people. So you don't need to be looking at a screen. The real world is your visual stimulus. So we can actually sort of get out of the lab and have highly controlled timing, but of the real world. We can flicker these at any frequency, so we're really not limited like we are on a screen to multiples of the refresh rate. We can really have microsecond level precision control over when the glass goes dark and light. So we can flicker at very precise frequencies. And this is completely mobile. This can be combined with a mobile EEG that attaches to the head, and people can sort of freely walk around. So what I'm going to show today is a few studies and some pilot data, just to sort of broadly illustrate how I'm using this method in my research and how it can sort of address some of the limitations that I just mentioned. So firstly, I'm going to talk about, just to show you, here's a little animation just to illustrate how we're making these SSVPs. So the glasses are flickering light and dark. In this case, it's at 10 Hertz. Obviously, it's slowed down for the purpose of the animation. So that is a square wave signal, which is driving the glass, which is when it's on, it's making it go dark, and when it's off, it's making it go clear. And this is also the trigger that we use to segment the EEG. This is the ongoing EEG. In this case, it was 10 Hertz, so every 100 milliseconds, we're taking a 100 millisecond segment of data and averaging them just to get the evoked potential, just like you would with an ERP. But obviously, we're going to have way more trials than an ERP. So this is flickering at 10 Hertz. So we would have 10 every second, 600 in a minute, 3,000 in 5 minutes. So we get very clean signal because we're averaging many, many segments. So this is what most of the data that I'm showing you. So the first thing we see is, so we start trying this. We have the mobile EEG and we get people out into the real world. And in this case, the real world is just the corridors out in the psychology department. That's as far as we've got so far, but in principle, this could go anywhere. And we start to see differences in the phase and the waveform shape of the SSVEP, depending on what people are doing, so whether they're standing or walking, and what they're looking at. So the most striking difference we see, the most obvious effect, is that there's a huge difference in the waveform shape of the SSVEP, depending on where the person is looking. So this person, for example, is either looking out of a window, and this is just 10 seconds of data, so he's standing there, looking straight ahead. The flicker is going for 10 seconds, so 100 segments of data average, and you get one specific waveform. And then when he's looking in a different place, standing just around the corner in a corridor, we have a completely different waveform. So there's a huge difference between the signal we're seeing, depending on where he's looking. And this is kind of highly reliable. So we can have somebody standing. These are pictures of what the participant is looking at, and each of these different visual streams gives us a distinct waveform shape. And when people come back later in the day, or even on the next day, what we see, the two lines here, is the same SSVP. So this is not just something that's sort of varying randomly, this is actually containing information about the scene. And this is actually good enough that we can have a distinct waveform shape, so we can decode where someone's looking. So somebody can stand in each of these locations for 10 seconds, and then come back the next day and stand in one of the locations, and we can predict with actually very high accuracy where they're standing, and decode that purely based on the waveform shape of the SSVP. That's a huge amount of information, really just from 10 seconds of data, which is much more than you get from 10 seconds of resting EEG, for example, or even from an ERP. So another pilot study we've done recently is to try and test this with eye tracking, so we can combine it with eye tracking. We have the eye tracking cameras can be attached behind the LCD glass, and I saw you have a front-facing camera that shows us what the participant is seeing as they move their eyes, and we can see what they're looking at different points in time, and then we can separate out the segments, so to create SSVPs depending on what someone's looking at. So somebody can be looking at a person, and we can take the flickers from there and create an SSVP of that, and then they can be looking away from the person, and we can create an SSVP depending on what they're looking at. And what we did in this first study, we tried to sort of just create a more naturalistic social situation. This is the front-facing camera, so this is what the participant is seeing, and they're just freely moving their eyes left and right, either looking at one person or another person on the left or the right, or just one person and then the empty space where the other person would have sat in another condition. And then in the final condition, there was just the same situation, but no people, and they were looking left and right to where they would have been standing. And again, this is 10 Hz flicker. And what we can see here, again, this is really just sort of pilot data to give you an idea, but the red and the green lines are looking at one person and looking at a different person. But that's both giving us quite a distinct waveform shape, and a very distinct waveform shape and phase depending when they're looking not at a person, even if there's a person in the peripheral vision. So this is sort of the first indication that maybe we can start to tap into the so-called third visual pathway, the sort of visual pathway in the brain that is dedicated to processing social interactions and biological emotion. Okay, I'll move on quickly. So the other point I mentioned that it is mobile. So in another study we had, we wanted to know the difference between people standing and walking. So we just had people standing and walking in a room where we illuminated the room with an adjustable spotlight such that the left or the right side of the room was illuminated. And in one condition, they were just simply walking across the room, looking straight ahead. In the other condition, they were standing at various points where they would have been walking to control for the visual input. And we wanted to see, can we get a clean signal from a single electrode, even with all the additional noise? Because the biggest problem with mobile EEG is all the additional noise that's caused from muscle artifacts and eye movement artifacts whenever anyone moves. So the way we tested this is we used a signal generator, a little device that generates a little square wave electrical signal that runs across the scalp that creates a sort of fake EEG, simulated EEG. So this is the EEG and the accelerometer reading from when someone's standing and when they start walking, and you can clearly see when they start walking, it's a really noisy signal. And we add this signal generation, then you can see the square wave pulse that continues throughout the EEG, even when they're walking. So we're putting the same data in to when they're standing and when they're walking, and then we're going to average these as if they were SSVPs, and we're going to see if we can recover a clean signal if we average enough segments and average all that noise away to zero. And the answer is we can. Here, if you average enough segments, eventually the walking and standing condition look identical, and that's when we know we have enough segments. That's how many segments you need to average to get a clean signal. So you can see as we increase the number of segments, the difference goes down to zero, and then approximately 2,000 segments is enough to say, with this amount of noise, the amount of noise we have from walking, we can really get a clean signal and that we can trust it. Yeah, that's a picture of the signal generator that we attach to there. So once we know we can get a clean signal, we can look at some real data. Here on the right is just an example SSVEPs from one person. What we're seeing is very different SSVEPs depending on whether the left or the right side of the room was illuminated, just like we saw with the different visual scenes. And what we saw at the group level is a reduction in amplitude when you're walking in the right parietal electrodes, but not in the left parietal electrodes. And we think that's to do with the additional inputs of the vestibular system. It's known that in right-handed people, vestibular inputs are processed primarily by right parietal cortex. And as you're moving, you're processing your own movement as well as just the visual scene. So we think this sort of lateralization is partly due to that. Okay, so the final advantage of these glasses, the thing that's really cool about them, is that we can flicker at any frequency, which means we can go through sweep through the entire sort of frequency range that we're interested in. So here's my resting alpha in the blue line. This is, we're showing the typical sort of peak at 11 hertz that you can see. And in the green is wearing the glasses, flickering at 5, 5.5, 6, 6.5, going up through the alpha range. And here we're just looking at the amplitude of the SSVEP, and as we track this, we can create a sort of amplitude envelope. We can see that obviously SSVEPs are higher whenever we sort of get close to the natural endogenous frequency, and this could be due to a number of reasons. It could be due to entrainment, or it could be due to a sort of resonance phenomenon. But typically what we see is some kind of, well, in some people, a relationship, a sort of correlation between the individual alpha frequency. In other sources, we don't see such a strong signal. But the key point is that there is frequency specific. So really, there are optimal frequencies at which we can be driving the brain. And this precise frequency is important. And that can tell us a lot about the underlying time constants of the neural circuits that are contributing to this. Yeah, so this is just an illustration of how we can do the very precise, here we did visual flicker at 20, 20.2, 20.4, we can really have precise control. And obviously, everything I've shown you so far has been in the alpha band, but we don't have to flicker in the alpha band. We can also go up to the gamma band, which is particularly interesting. It's a much smaller SSVEP, but again, if we have enough trials, we can get a clean signal regardless of movement. As I mentioned before, we do see interesting differences in the gamma band that we don't see in the alpha band, such as phase shifts related to when people are moving. And again, we can create these sort of alpha amplitude envelopes throughout the gamma band. So here is the, again, we're flickering at 20, 25, 30, 35, all the way up to 60 hertz, or 55 hertz, and we see how the amplitude varies. The blue and the red is two different visual scenes. So like I showed you before, somebody's either looking out of a window or somebody's looking across the room. We see a similar pattern across in the amplitude envelope. So there are certain peak frequencies which are consistent depending on the individual. And lower frequencies at parietal electrodes, we start to see, like we saw before, a distinct waveform shape and phase depending on the visual scene, which tends to disappear as we get up into the higher gamma band. But the gamma band shows us interesting resonance, which is not always at exactly 40 hertz, which is important because when people are often studying the gamma band, they often just flicker or drive the brain at exactly 40 hertz, but actually there's individual optimal peaks there as well. So I'm out of time, so I'm just going to say, yeah, the long-term goal really is to study the effect of body, eye, and head movements. So what we want to do is use this in all kinds of types of movement that we would naturally see in natural exploration of the real world. So eye movements, head movements, running, standing, jumping, and anything you want, and any combination of these. And also we want to study the visual processing of real visual scenes and social interaction and sort of start getting neuroscience out of the lab. So yeah, that's my website. Get in touch if anyone's interested in using this method or just learning more about it. And yeah, thank you.
Thank you, James. So as for the news talk, if you're watching this and you're interested in what James is doing, please do get in touch with him and ask any follow-up questions. An extra round of applause for our 2 speakers in this symposium is in order because they were speaking while the world is ending outside. So, yep. That concludes the online part. Thanks for watching, and please stick around if you are here.